extends below that all the way down to the corpus callosum, which we're going to get to in a second.
Yeah, well, ours is the spinal cord.
So now we want to focus for a moment on the gray matter and white matter just as a general rule, and we're going to see those several times as we go forward. So when you hear gray matter, what we're talking about is where the nerve cell bodies are located. White matter is where nerve fibers are located, and it's usually termed white because they're myelinated. That's why you can tell. Okay, and so basically those in the brain. Areas of myelinated white fibers are called fiber tracts. Outside the brain, they're called nerves. Okay, they're the same thing because there are a whole bunch of axons. And so let's look at. Let's go back to this guy for the moment. And so this is a section, and again, we're just going to use this to illustrate gray matter and white matter for the moment, and then I want to go to the whole brain over here. So the area right in here, this is a part of the cerebral hemisphere. It's part of the cerebral hemisphere. This is the inside of the lobe. Okay, so they're not connected. I haven't cut those at all. They're separate. Okay, and so the area up here, irrespective of the pia, this is where all the nerve cells bodies are located. If I go down farther, you see it gets wider, and then down in here, these areas right here and here are fiber tracts. These are axons. Okay, and some of these, depending on their preservation, they show up as whiter or less white. But the point is, we'll see those momentarily. Now, as you go down into here, this area right in here, this last extension is the spinal cord. And you have white on the outside, you have gray on the inside. So the gray matter is on the inside of the spinal cord, but it's on the outside of the brain per se. You almost lost that one. And then the best place, probably the easiest place to depict and visualize gray versus white is by looking at the cerebellum. Okay? So you see, looks like a little tree, doesn't it? Isn't that cute? Okay? Galen called this the arbor vitae. Galen was the greatest, Greek anatomist that lived. He made most of the structures. It's still around today. The arbor vitae, the tree of life. Okay? So this is we see this in the cerebellum, and the core here, the white area, is fiber tract, and the little brown areas, the leaves, if you want to call them that, that's where the gray matter is located. Nerve cell bodies are located there. Okay, so it's just an example of what that looks like. Now it's the same thing over here, they're just different color because of the preservation.
Go ahead, Mike.
Okay.
The lighter parts,
the lighter parts, the gray matter, myelinated, absolutely. The dark parts are nerve cell bodies, and to be honest, unmyelinated fibers. You can't differentiate. Okay?
Is that because it's the fatty?
Fatty. That's what shows up.
Makes it white?
Absolutely. Yes. Absolutely. You got that. 100%. Okay? So now we're going to look at some brains. Where did my brains go? We're going to look at some brains that have the dura taken off, and so we want to look at the major structures, and we'll look at from the top and then from the bottom. So basically the major things that you see on the superior side are these two big lobes here. They're separated manually here, but there's nothing that manually separates them. They're connected and pulled together by the congealed arachnoid matter. Okay? This is the cerebrum, and these are the cerebral hemispheres. Right and left. Cerebral hemisphere. Half of the cerebrum, they don't look like, same roundness as us, but that's okay. So basically, that's what you're looking at there. And then posterior to those is the cerebellum. Okay? Now, because we're bipedal, this is turned at a 90-degree angle in humans. Okay, that's all. And then posterior to that is the spinal cord. So it's all continuous. Okay? You have much more white matter down here.
in humans with the spinal cord then be inferior instead of posterior?
Yeah. Yeah, unless you're lying prone. Okay? So now what I'm going to do here is if we flex this, and we can't, this is kind of damaging to the brain. What we're doing on this one's already broken more than the other one. So essentially, if we look at this, and basically what you're seeing down the bottom there, these are fiber tracts that are holding this thing together.
That's what happens in a car accident with
You laugh, yeah. Except that the difference would be that, you know, your brain's not this preserved, so it would actually, you know, just squash and go back to something. The other thing you can see here in this one is you notice this tissue that I'm pushing on right here. See that? That's the arachnoid mater. Okay? Looks like this elastic tissue. Pretty cool. So it surrounds the outside of everything. Remember, in life, CSF goes underneath that. Okay? So the reason I'm flexing this is so we can see two other major structures. I'm probably going out of order, but that's okay. These two things are part of the midbrain. There's 4 bumps right here. 2 big ones on the top, 2 narrow ones on the bottom. See those? 4 bumps. See it? Okay. Together, they're called the corpora quadrigemina, the 4 bodies, or the body of 4.
Oh, that.
Corpora quadrigemina. Okay. But that basically is a whole group. The reason we do that is because they're part of the midbrain, and they're responsible for auditory and visual reflex responses. What do I mean by that? So the 2 big ones on the top are the superior colliculi. They're associated with visual reflex response, startle responses.
The 2 big ones?
The 2 big ones, the superior colliculi. Okay? So what do I mean by visual startle responses? So you're focusing on something, and then you see a flash out of the side of your eye. You know, you're not really looking, and all of a sudden you look over. That response is mediated by the superior colliculi. Okay? The 2 lower ones are those mediate auditory startle response reflexes. So instead of a flash, you hear a bang, maybe a gunshot, you know, from behind you, and immediately you whip around. And that's mediated by these 2 inferior colliculi. Okay? So these are all part of the more primitive brain.
Are those mostly gray matter?
They're white and gray. So they're kind of mixed in here. Okay? When
I
you
guess
get
you're
it
going to have more white and gray in most
in a lot
areas.
of them. Yeah, the difference is when you look at the cortex, and this is a good lead in, because the cortex of the cerebrum, you see, is wrinkled, has these bumps and grooves.
And what is the cortex?
The cerebral cortex, that's where the nerve cell bodies are located. So all of your neurons in your cerebrum are in the cerebral cortex. Okay? When you get down into the white matter, those are all the axons from those neurons. Okay? And the reason that you have all these bumps and grooves, and by the way, the bumps or the ridges are called gyrus, or singular, and gyri, plural. The grooves are called sulcus, singular, or sulci, plural. Now, in these, where's this other guy? You see where it's nice and white here, you can see these dark spots in there. Those are blood vessels that were captured by the collapsing arachnoid mater. And you have blood vessels that run in those grooves. Okay? But the whole purpose of the grooves and the bumps are because if you spread out the cortex, then it's about a 1/4 inch thick, and it's spread out in humans, about the size of this bench.
Wow.
And how do you get in there? You put it in wrinkles, grooves and folds. Yeah, that's literally what it is. Origami for your brain. Okay, and that's significant because in humans, not in sheep, we've actually designated, figure out what those individual gyri do, and that's when you look at that one, psychological brain model. It has everything written on it. Precentral gyrus, and your fingers, toes. And how do we do that? How did researchers do that? They cut a person's skull apart, opened up their, dura mater, and stuck little probes in there and said, "What do you feel?" And they told them. Yes, absolutely. That's exactly what, you're laughing, I'm not joking.
Absolutely not.
And the other thing is, that's how they figured out what sensations were. That's how they figured out the mapping of the precentral gyrus, or the postcentral gyrus. And then with the precentral gyrus, they did the same thing, and they would probe that, and the guy would stick his finger up. Or they'd probe another one, he'd stick his tongue out. Or they'd probe another one, he'd make a noise. And that's how they figured by doing that. And so you say, well, how come you couldn't feel it? Because your brain itself doesn't have sense organs to feel pain. So you can stick all these other things in there, and they can do all this stuff and ask you questions, and
Wow.
that does
That is
happen.
crazy.
What would they use like criminals? I mean, I'm sure people.
No, it's oftentimes this happens because of